Hello everyone, welcome to Toppr.com. So today's session will be for biology and it is a live class session. We know that we are conducting these type of sessions every week and there are many subjects we are covering in this type of live class sessions. So myself Pushpendu, I'm a subject matter expert in biology in Toppr.com. So today we are going to talk about the topic biomolecules. Right, so we are going to cover this topic biomolecules, and let me give you a brief about how are we going to conduct this session. So in this topic there are many sectors where we can actually go into a lot of depth in this topic, but here in today's session I'll try to give you a brief idea about all the biomolecules that are present and that is necessary for you to understand. What are the areas you must concentrate on, what are the things you must understand. We'll talk about those factors, and also we'll talk about a very important part, that is enzymes, right? So let's start with this. Before we start, I'll request you to just follow us on YouTube, subscribe to our channel, keep asking doubts while we are conducting this session so I can try to solve those problems, and even if time does not permit within this session, keep posting the doubts, we'll surely get back to you. So let's start with today's session. So today we are going to talk about biomolecules. So from the name only we understand what does this signify. So this word biomolecule, that means something related to life, correct? And molecules, something which is helping in creating this life form, correct? So these are the molecules which help to create the life forms. So initially, if you just talk about the history of how life has evolved, there are many theses or many theories behind how life has evolved. We'll see there are many experiments which has been conducted in which scientists thought that these biomolecules were formed initially and they combined, they collided with each other, which actually resulted in the formation of a life, that is the cells, initial cells. Okay, so yes, so biomolecules are one of the most important components for the cell, which results into the formation of life. So what are the biomolecules? I'm sure you must have heard about few components of biomolecules that we are going to discuss today. Let's see what are they. Carbohydrates, right? I'm sure you must have heard about these words. Carbohydrates, proteins, fats, vitamins, and also minerals. So these are some of the important components which are required for our body. And also if you talk about nutrition, these are some of the important nutrients that we require for our body. So हमारे body के लिए ये nutrients भी बहुत ज़रूरी है. So can you just name from where do we get carbohydrates? Carbohydrates कहाँ से मिलते हैं? For example, potato, right? Rice. These are the sources of carbohydrates. What about proteins? Dal. This is one of the areas, this is one of the pulses, mostly the pulses, it gives us proteins, correct? Egg, the meat, if you talk about fish, so these are the sources of proteins. Fats, mostly the oils, butter, ghee, so it has a lot of fat content which are essential. Vitamins we can get from a lot from the fruits. Minerals, of course, there are from the fruits also we get a lot of minerals. So these are the sources which are important for our body, and the interesting fact is that these are the molecules which help in constructing our body. Let's see how. So first we'll try to cover the structures of these type of molecules, how they form. Okay, let's talk about carbohydrates. The basic unit of carbohydrate, if we talk about, is what? Glucose. We know this, right? So if we try to draw the structure of glucose, I'm sure you must know this, but let's try to draw it. So how many carbons are there? 6 carbon molecule. Just try to understand how I'm trying to draw this figure, right? So here this is a cyclic structure, and here we have OH, H. So let me just draw the structure over here. Same. So these are all the bonds, H and OH at each carbon. So these are the carbons. here also OH, and here we have CH2OH, correct? Now, so can we name the carbon, can we number the carbon? So this is the first carbon. You have to remember this so that when later on जब हम बात करेंगे about bonding, when we talk about bonding, how polymers are formed, multiple units combined together to form a big molecule. So these we need to understand. So this is carbon number 1, right? This is carbon number 2. Here this is carbon number 3, 4, 5 and 6, right? So 6 carbon molecule, we know glucose is a 6 carbon molecule, and this is a cyclic structure. So please take a note on this structure, how it looks like, because this will be needed. And you must have heard about like alpha D glucose, right? Beta D glucose. How these are formed? These are nothing but the orientation of these molecules. How these molecules, how this OH at each carbon is oriented, or the stereochemistry you can talk about. The stereochemistry determines how the stereochemistry orientation of the molecule is like. So there are different forms. Let's not talk about this at this moment. So we'll just understand this is the basic structure of glucose, or the basic unit of sugar, you can say. So these glucose molecules, they combine together to form the carbohydrate. So glucose is the basic unit. Now, so this I've talked about the carbohydrates. What about the proteins? Can you tell me like what are the basic units of proteins? They are amino acids. Amino acids, I'm sure you must heard about this terminology, amino acids. So amino acids combine together to form the proteins, right? Now there are 20 amino acids, right? So we can denote amino acids in this abbreviated form. There are 20 amino acids, and you would be surprised to know that the combination of these 20 amino acids makes the all different types of proteins that is essential for our body. And in fact, when you later on study more in depth about biomolecules, about biochemistry, you'll understand that these protein molecules are very, very essential for our body, for each of the cells to function. These proteins make the channels through which the inner and the outer side of the membrane or of the cells interact with each other. Okay, so the protein forms the channels. The proteins are the components of enzymes, and I'm sure you know how interesting or how important enzymes are for our body. Without the enzymes, we cannot stay alive. Not only us, none of the organisms, even if you can talk about the single-celled organisms like bacteria, they also cannot survive without the enzymes. So proteins are the building blocks of the enzymes. So enzymes are made up of proteins. Now fats, similarly vitamins, minerals. So these are the important biomolecules. So let's see about or talk about the bonding patterns. Now before I talk about the bonding pattern, I'll just mention two important areas or two important terminologies. Metabolites. What are metabolites? Metabolites, they are nothing but these biomolecules. Okay, these small molecules, they form the metabolites of the cells. So let's talk about the cells. We are not talking about the body, we're talking about each of the cells. So metabolites, they are part or the minute structure that is present inside the cells. They combine together to give higher complex molecules. So metabolites are the biomolecules. Now metabolites can be of two major types. What are they? Primary and secondary. So दो different type of metabolites हो सकता है, primary and secondary. So what are they? Primary metabolite means these are metabolites which are actually have functions. In fact, so that means, does that mean that secondary metabolites do not have function? Then if that is so, why it is present? So let me explain you. Primary metabolites, the function of these metabolites have been discovered. We know the exact function, and in fact, if I put it properly, the function of the primary metabolites is visualized. We can easily observe the function of the primary metabolites. So what about the secondary metabolites? These are the metabolites whose functions are yet to be discovered, or whose functions are known but they are not needed to support the life. hence they're known as the secondary metabolites. So you get it? Primary metabolites are the primary ones which are required life support करने के लिए, उस organism या उस cell को जिंदा रखने के लिए, उसको live रखने के लिए ये primary metabolites की जरूरत है. They need primary metabolites, whereas the secondary metabolites provide, I state it as additional advantages. So let me explain you what are the additional advantages. For example, there are certain fungal cells which can secrete antibiotics, correct? So antibiotic can be referred to as a secondary metabolite. Why? Because antibiotic, that means it is against bacteria. Why would fungus secrete or synthesize something antibiotic inside itself, right? So this does not have any direct function, but it helps in its defense mechanism. If there is a bacterial infection where fungus is growing, then it would help. So it is a secondary metabolite. So I hope you have understood the difference between primary and secondary. So this is important. So let's move ahead with the discussion. So please keep posting your doubts. Please ask questions so that I can help you understand better. Invite your friends so that they can also be helped by our sessions, right? So, okay. Here we have talked about the metabolites, two different types of metabolites. Now let's talk about the bonding patterns, how this bonding actually happens in different areas. Can we talk about the first bonding? Let's talk about the carbohydrates. Let's see. Again, draw the basic structure of the carbohydrates. I'm not drawing it fully. Let's just mention the OH. That means the opposite will be H. So here I'm mentioning the OH. Correct? Now, here what will happen is how this molecule will be joined with the next one. So let's see how this joining actually is happening. Okay. So I hope you can relate the structures. These are the carbon. So these carbohydrates, you understand, this is a complex structure of the basic unit of glucose. So this is a glucose unit, this is another glucose unit. So based on different types of bonding, there can be different forms of carbohydrates. Like we have sucrose, we have fructose, we have saccharose. So all these are different types of carbohydrates. How they are different? Because they are all made up of the simple units like glucose. The thing is, the bonding patterns are different. Now, if you again recall about the number, this first carbon, second, third, fourth, fifth and sixth. First, second, third, fourth, fifth and here will be sixth. So now the bonding can be 1,4 glycosidic bond. It can be 1,6 glycosidic bond. So this bond, as I've mentioned, is known as glycosidic. Let me write it here. Glycosidic bond. Okay. So let's see how this glycosidic bond is forming. Now here what happens, for example, if we talk about this 1,4 glycosidic bond, what will happen is, here is also we have OH and H, CH2OH, correct? This is a dehydration reaction. So इससे क्या समझ में आ रहा है आपको? Dehydration मतलब पानी निकल जाएगा. Water will be expelled out. So dehydration. So can we see? Let's try to dehydrate this end 1 and 4. So here you are seeing there is one OH, there is one OH. So from here what we need to take it out? From here you have to take out one molecule of H2O, correct? Then only it will be a dehydration reaction. So now the bond, if I draw it, should look like this. Okay, so here we have one molecule of glucose. This is the next molecule, correct? Now the bonding would look like somewhat. Here it would look like, that means one H will go and one O will go and another H will go. So it will be bonded by this. You understand? I'm not drawing the other part. So here we have the normal bonding that is there. So this is the glycosidic bond. You understand? From here, let's imagine here H and O has gone. This H is expelled out. So that is H2O that combines to form water molecule, a molecule of water. So this is a dehydration reaction, and since these are forming the sugar, they are known as the glycosidic bond. Okay, so similarly, let's see what happens while the formation of amino acids. Okay, amino acid, they have a basic structure. If I represent the basic structure of amino acids, they are represented. They have two forms, like they have two major areas. This is the carboxylic group, and they have the amino group. Carboxylic group and amino group. Now let's see how they are bonded. Again, and of course carbon will have 4 bonds, so this is the functional group. Now I've said that there are 20 different amino acids. How they are different from each other? This is, if I just remove this R, this is the basic skeleton of each and every amino acid. Every amino acid differs from each other just with this. Okay, this functional group. This group, R group, actually varies, which forms different or 20 different types of amino acids. This is the basic structure. Now let's see how this is bonded. There is a nomenclature which is followed every time when we try to represent. Let's write it R1, R2, so 2 different amino acids. Whenever we try to write an amino acids, the amino group is always kept towards the left hand side. Okay, this is left hand side, this is right hand side, and again left hand side, right hand side. Now what happens? This is also a dehydration reaction. Now I'll just give you few minutes and you think about how can some bonding occur. It can occur in this area, this carboxyl group and this amino group. Just think about it so that I can represent it, right? I'll use the next page and we can see how we can represent the bonding. So take your time to think. By the time, I'll just try to draw the molecules. So here we have the 2 different amino acids, correct? Now let's see how this is bonding. Again, I have said that it will be a dehydration reaction. So let's write it down. Let's see what is happening over here. So this is R1. Here we'll have CO, NH2, and here we'll have CH, R2, COOH, and now we have NH we bonded here. Okay, so the reaction will be somewhat like this, correct? So what type of bonding this is called? This is known as the, because during the time of the carbohydrates it's known as glycosidic bond. Here they are known as the peptide bond. Okay, they're known as peptide bond, correct? So just take a look how this reaction has happened. Peptide bond, right? So just take a look how I have tried to represent here how the reaction has taken place, correct? So again, let me just repeat it. Here we have the amino group, here we have the carboxyl group, dehydration reaction, water has gone, and this will be the bonding pattern, correct? If I just represent it properly, it can be represented as, here carbon will be double bonded with oxygen, right? Oxygen, you know, it can do double bonding, so valence is 2. So here double bonding O, here H, and CN, it is a CN bond, carbon nitrogen bond, correct? So we have represent the peptide bond. Now in this way this will continue and it will form big polymers. Okay, so I hope you have understood how peptide bond and how glycosidic bonds are formed. Now, in a similar way, you've heard about DNA, right? Deoxyribonucleic acids, DNA, how they are formed, what are the basic units? DNA has 3 basic units. What are they? The sugar backbone, right? They have the bases, they have the ribose sugar. Let me represent the ribose sugar, how it looks like. The ribose sugar looks like this, okay? Here, the base is attached. We have 4 bases, you know, like adenine, guanine, thymine, cytosine, and also uracil, correct? So, right, base, and here we have the phosphate group, okay? Here we have the phosphoric acid, which is phosphoric acid which is attached, correct? Now we have talked about this peptide bond. Now I'll talk about few things which is very essential for you to understand, and these are the areas where we get confused. The 3D conformation of proteins. So these peptide bond together, many peptide bonds will form a polymer, and that polymer will result into the proteins that we are talking about. Now the proteins have different structures. They have the primary structure. What is the primary structure? Now this is the primary structure. We can call this as a primary structure of the protein. Now this primary structure, that means n number of amino acids can join together to form a linear chain of primary structure. Now when you understand this linear chain, which was like this, if it starts coiling, if it starts forming bond and it forms a coil structure, that is helix, this is called a helix structure. It forms a helix structure, then this is called a secondary. This is called the secondary structure of the proteins. Now primary, secondary, we have tertiary. What are the tertiary structures? Now these, we have seen some bonds over here. These are nothing but simple hydrogen bonds. Now what happens here? There are certain disulfide linkages. So if you study in more depth into these, you'll see there are disulfide linkages, and these proteins which are already coiled, they form bonding with each other. So you understand, this is the coiled structure which is now forming this type of structure. So this is called the tertiary structure. This is actually the 3D conformation. This is actually the 3D. And tertiary structure is the 3D conformation of the proteins. Now, do we have anything higher than this? Yes, we have. We have the quaternary. Quaternary structures. Now, what are they then? So this is the 3D structure of a particular subunit of a protein. It can be. Now, some proteins are there which are formed of many different subunits. So those form the quaternary structure. You understand? Different types of 3D structure, they fit together into different domains and they form the quaternary structure of the protein. Now, let's talk about anything else. Yes, we have 2 important terminologies like anabolic and catabolic. Anabolic, what are anabolic reactions? What are catabolic reactions? I can help you out. Like I remember these as catabolic. I refer this as cut. Catabolic, that is referring to a cut. That means it's breaking down reaction, breakdown reactions. Anabolic will be the opposite. That means joining reactions. Anabolic से बड़े molecules बनते हैं, catabolic से बड़े molecule तोड़ के छोटे molecules बनाया जाता है, right? So these are the 2 types of reactions. Now the important topic that we need to discuss is about enzymes. So what are the enzymes that we are talking about? Enzymes are the biocatalysts. Biocatalyst, okay? Now why this is important? Why we should study about enzymes? Within a body, whatever reactions, whatever metabolic reactions are taking place is taking place with the help of enzymes. So enzymes are very, very important for us. Now why? For any reaction to happen, we need a particular threshold energy that is required for that reaction to take place. So I repeat, कोई भी reaction होने के लिए एक particular energy चाहिए उस reaction को complete करने के लिए. Now enzymes play a very important role here. अगर कोई लेट्स इमेजिन रैंडमली 10 यूनिट्स ऑफ एनर्जी कोई रिएक्शन करने के लिए ए टू बी, ए को बी में कन्वर्ट करने के लिए, if we require 10 units of energy, the enzymes can they bring down this energy requirement to about 2 to 5. You understand? More than half. It can bring down the energy requirement. So you understand this is very energy efficient. We do not require a lot of energy in order to perform all these reactions, and we know that within our body there are many reactions that is happening to keep us alive. हम लोग जिंदा है इसी वजह से कि हमारे बॉडी में हर मोमेंट, हर सेकेंड, हर माइक्रो सेकेंड बहुत सारे रिएक्शंस कैरी आउट हो रहे हैं. सो एंजाइम्स प्ले अ वेरी इंपॉर्टेंट रोल. एंड सबसे इंपॉर्टेंट पार्ट एंजाइम्स के ये है कि एंजाइम्स आर रिसाइकल्ड. हाउ? ये जो रिएक्शन है ए से बी टू फॉर्म बी फ्रॉम ए, द एंजाइम टेक पार्ट in this conversion but itself do not get converted. That means A will transform into B, but after the reaction E will remain E. There is no change in the enzyme structure. That is the specialty of the enzyme. Now let me represent a very important energy curve for the enzymes which we should know. So I'll represent it graphically. So here the reaction, how the reaction is progressing, and here will energy, potential energy. Okay, now let's represent. So there are two things we know there are substrate and product. Substrate, so for this reaction A is the substrate, it is converted into B, which is the product. So for example, a substrate A, if it normally requires this energy to form the product, so here we have A, here we have B. You understand? So this is the amount of potential energy which is required to form this product. But if we use enzymes, this can be reduced to this. This is enzyme modulated, okay, enzyme plus. So this is basically the reaction or energy required for the enzymes. If it is an enzyme catalyzed reaction, then it will require this amount of energy. If it is not an enzyme catalyzed reaction, then the amount of energy, that is threshold energy required, will be much more. Okay, so this is known as the activation energy. So the threshold energy that I was talking about is the activation energy. That means ये enzyme जो रिएक्शन है ए से बी कन्वर्ट करने के लिए जो एनर्जी चाहिए उसे कहते हैं एक्टिवेशन एनर्जी और द थ्रेशोल्ड एनर्जी फॉर द रिएक्शन. ओके, सो दिस इज द एक्टिवेशन एनर्जी विदाउट एंजाइम्स, एंड यू सी हियर this is the activation energy. Let me use a different color. So this is actually the activation energy. The green part is just the activation energy with the enzyme. So if you compare, the activation energy required for a reaction to take place without the enzyme is much higher than that of the energy required with the enzyme. So you understand how enzyme is important, and the important part is this enzyme will remain as it is. After this reaction, enzyme is just in the same manner or similar manner, and it is ready to form the or to take part in the next reactions, consecutive reactions. That is, enzymes are recycled naturally. Now there are certain factors which are affecting the enzyme activity. Okay, let's talk about quickly the factors which are essential for the enzyme activity. Enzyme activity. What are the factors? Okay, first of all, temperature and pH. All enzymes act at a particular specific temperature which is known as the optimum temperature and optimum pH. For example, most of the enzymes which is present in our body, they act at an optimum temperature of about 37 degrees centigrade. Okay, so that is the normal temperature or room temperature. Okay, certain enzymes act a little below, like 25 degrees centigrade, which is the room temperature. Now there are certain enzymes, for example, you'll study when you in your 12th class, when you talk about the rDNA technology, we'll study about Taq DNA polymerase. So this is an enzyme which helps in the polymerization reaction of the DNA, and it can work even at a temperature of 120 degrees centigrade. And the structure of the enzyme will not be ruptured at 120 degrees centigrade also. Whereas the enzymes present in our body, they can get ruptured even in 40 degrees centigrade. And this is the enzyme I'm talking about that is found in a bacteria. Okay, so this is very important for us. Similarly, the pH. The pH, all the enzymes act at an optimum pH. Some of the enzymes are neutral, that is at a neutral pH, that is a range from 6 to 7, they can work well. Some are acidic pH, they work well in acidic pH. For example, enzymes which are present in our stomach. We know that stomach acidity is about 2 to 2.5. So those are the enzymes which is present in stomach. They are acidic in nature. They're not acidic, actually they work well in acidic type of environment. Whereas the enzymes which are present in the intestine, because of the presence of bile juice, they are mostly basic in their reaction. So they like basic environment. So the enzymes will work if you just try to plot it through a graph. The graph will be bell shaped for both temperature and pH. Okay, if I pH or temperature, and here if I draw the activity of the enzymes, then it will be bell shaped. That means this is the optimum range. This is the optimum range where it will work maximum, right? So temperature and pH are the important factors. Second important is the substrate concentration. Okay, how much substrate is there? Now, there's an interesting phenomena which has been observed is with the increase in substrate, the enzyme reaction increases, but after some time it comes to a saturation point. Why? Because all the active sites of the enzyme which is responsible to perform the reaction, they are occupied. So after some time it becomes saturated. Okay, it increases exponentially very fast and then it becomes saturated. So temperature, pH and the substrate concentration, these are the important factors. Also I'll talk about one of the terminology, that is inhibitors. What are inhibitors? There are certain molecules of enzymes which are similar, which has similar properties like that of the substrate. So enzymes, they bind to the substrate, correct? Enzyme, for example, if this is an enzyme, this is the substrate, it will bind to the substrate. But there are certain inhibitors which mimic these type of substrates. So they are called inhibitors because they inhibit the functioning of the enzyme. They will bring down this reaction rate. So these are certain factors which help which actually determine the enzyme activity. So in today's session, we have talked about all these basics about biomolecules. Now we can talk about a lot in depth about these. You can go through many different books, NCERT or different books, different reference books. Also you can go through like ABC of biology, you can go through Truman's. So these are the books which will cover a lot of details about this topic that we have already discussed about. So if you have questions, quickly ask. I can try to help you out with the questions, answers, and in the meantime I'll just revise what we have talked so far. So here we have talked about different types of molecules, right? We have talked about the different biomolecules, that is carbohydrates, proteins. We have talked about the amino acids. We have talked about nucleic acids, right? We have majorly covered enzymes because enzymes are very important. We have talked about the activation energy graph. Okay, that is usually asked in your examinations. So activation energy graph, that is very important. Activation energy, what is activation energy? What is the main function of the enzyme? What are the factors we have talked about that helps in determining the enzyme activity? And also very importantly, inhibitors. If you just do a bit of research about this inhibit activity, you'll see there are many different types of inhibitors also. Okay, so it results into competitive inhibition, non-competitive inhibition. So these are the different types if you go into depth. So that's all for today's class. Quickly, if you have questions, ask me so that I can give the answers. Okay, so I've got a question that why amino group is written towards the left. So this is a nomenclature which is followed, like there's no proper rule that why that is so. That is a standard form which is followed. Like in chemistry you have like IUPAC nomenclatures, so there are certain standard things which are followed all over the world. So that is a standard which helps in representing your bonding, okay, so that you do not mix up. So that is a general trend in which we are supposed to represent. So let's see if we have other questions. So I think that's all for today in this session. So just let me take a look if we have another question over here. Okay, so you're asking about an example of competitive inhibition. Okay, I can say you, but it will be a bit unrelated to this here. Right now, what I remember is about, if you talk about if you remember what is happening in glycolysis reaction, you remember that the carbohydrate, right, glucose is broken down to pyruvate. The first reaction, if you remember, glucose to glucose 6-phosphate. So in that reaction, hexokinase is used. So that acts, that that is a competitive that enzyme can be inhibited competitively. Okay, so there are many reactions which actually can be inhibited in this. In fact, this is a area in which you'll see many diseases are actually, we are trying to cure those diseases by this technology. If there is a disease in which a certain enzyme is acting too much, then what we do is we try to inhibit the activity by mimicking that particular substrate, you can say. It can be done synthetically also. We can make synthetic substrate inhibitors also, right? So yes, there are many examples you can talk about. So I think for today's session, that's all for now. So keep asking these questions. We can surely get back to you. And do remember that we are conducting this type of sessions every week. And in different subjects, we are covering these type of sessions. So you subscribe to our channel, YouTube channel, invite your friends so that they also get help from us. And keep following our channel. We are doing these type of many sessions. We have a lot of sessions which is going on over here. So I think it's very helpful. We are getting a lot of good feedback from you. So that's the reason we are continuing. So thank you so much and thanks for joining us.